All right, everyone. It is my pleasure to welcome you to our next session, Tools for Building the Synthetic Biology-Based Economy. I'm Ben Brown, and I will be, have a joint appointment at UC Berkeley and Lawrence Berkeley National Laboratory. And it is my pleasure to introduce Eduardo Abeliuk, who is a visionary working at the interface of AI and synthetic biology. He's trying to decode and then design life to spec to accomplish specific aims. So Eduardo, please introduce yourself and tell us a little bit more about how you came to work in this area and what is this area?
Sure. Hi, Ben. Thanks for the nice introduction. Well, a little bit about my story. My journey began, studying physics and electrical engineering. And although my PhD was formally in electrical engineering at Stanford, I was a member of the Information Systems Lab, which is the group of people at Stanford that are interested in algorithms and for information processing, machine learning, and math applied to a broad range of applications, including not only electrical engineering, but also computer science and biology. And I've always been interested in science and understanding data. And that's where I basically started, on one hand, teaching myself computer science and biology. And I met the founding team of Teslagen at Stanford, my colleagues Nathan and Mike. The three of us overlap at Stanford, and we had, the three of us, a common background in physics. But we were all, the three of us, interested in biology and really experienced a lot of pain when trying to design biological experiments ourselves. I spent a number of years doing some basic molecular biology research. And we found that a lot of time is really wasted designing and running experiments. And a number of tasks can be automated, and algorithms can be trained to guide your experiments. That basically led to the formation of Teslagen. And back to your second question, what is synthetic biology? What is AI? Well, I would say it's not something like radically new, like the evolution of the web. People now are talking about the web 2.0, the web 3.0. For me, synthetic biology is like biotechnology 2.0. Biotech 1.0, if you will, was born a few decades ago, thanks to the development of a few key technologies that allow people to cut and paste DNA very effectively, and techniques to amplify DNA easily. What we know today as PCR, people have heard about it through the COVID testing. Then came the DNA sequencing revolution, where we could read DNA much faster and much cheaper. And then synthetic biology basically emerged from when a group of scientists started developing techniques not only to read DNA very efficiently, but also to write DNA. And since then, the cost of writing DNA has been dropping significantly. Like a few decades ago, it used to cost roughly $10 to synthesize one single nucleotide of DNA. Now it costs roughly 1 penny. The costs will continue to decline. And if on top of that you add capabilities to efficiently assemble DNA through a number of modern techniques, and also to edit DNA through the so-called CRISPR-Cas9 techniques and revolution, coupled to a new capabilities to train advanced deep learning models that can now be trained on the cloud very efficiently and guide experiments, then you have what I might be calling biotech 3.0, or synthetic biology plus AI. And the important I think here thing to note is that I think that all the portions of our lives are going to be transformed in one way or the other by synthetic biology. It's going to impact every sector. We're going to be, in the next 10 to 20 years, really transitioning into a bio-based economy that will touch, the healthcare sector, energy, the food industry, climate change, etc. So it's going to be really disruptive.
It is an incredibly exciting time. I mean, I obviously work at Berkeley National Lab, and there's a huge focus on bioenergy. And we've seen synthetic biology start to emerge, designer organisms to produce biofuels. But of course, now we've seen the emergence of the electric car and things like this. What do you think are the prospects for moving away from fossil fuels to a biofuel economy in the next 30 years?
Definitely, I think this is one of the areas where we are already seeing some impact. It's, for example, we work with a company called Lancatech based in Illinois, who is already optimizing some anaerobic microbes that eat carbon monoxide and carbon dioxide to produce high-value molecules such as ethanol. So imagine that microbes that can grow in large bioreactors installed next to large industrial plants and steel plants that generate a lot of waste gas, and that can not only be used to heal the planet, save it from climate change, but also produce as a byproduct a biofuel that can eventually replace fossil fuels. Also, the Department of Energy is now investing. They recently announced a new entity called Biomate that is through the Air Force. U.S. Air Force is funding a number of initiatives to push the industry to more effectively and efficiently produce, bio-based fuels. So there's a lot of, innovation and investments that are, going into biofuels.
So one of the challenges with bringing synthetic biology to bear in our lives and impacting society in a substantial way has been the slow turnaround time, right? There are some very important success stories, taking malaria medication out of plants and putting it into a bioreactor with yeast and a bacteria. Incredible success, produces 80% of the medication for malaria in the world now. But that one feat of engineering was 15 years, 2 decades of work. So what are the major challenges today that the biotech industry is facing, especially around acceleration? And how could advancements in AI help tackle these challenges?
Yeah, that's a good question. So I would say that at the highest level, of course, like the greatest challenge in biotech in general is, designing and developing new biological products much faster. It's still very hard, very expensive, but particularly very slow, to develop like these products, bio-based products. Speed, is what matters the most for a number of industries, including pharma. I don't know if the audience knows, but it can take on average 10 to 15 years, for example, to develop a new therapy, a new drug. And this not only affects pharma, as we were talking, it affects the entire biotech industry that seeks to develop a new generation of materials, biofuels, food, enzymes, microbes for industrial use in general. And I think AI can really help to develop different biological products in much shorter periods of time, basically by exploring, the search space of possible designs more effectively, thus reducing the number of iterations needed to optimize a biological product or a biological process. So, it can help to find that optimal solution faster.
So looking forward a little and say the next 10 years, where do you see things going in this sort of near term for SynBio?
Yeah, so particularly in the area where I'm working on, which is SynBio and the intersection between SynBio and AI and software, something that I see is that definitely there is a new industry that is emerging within biotech. Biotech is already a large market. Roughly $750 billion are spent every year on biotechnology. And the biotech market will continue to grow fast, but a more significant fraction of it will be spent on computational tools. So let me make some analogies with other industries that maybe the audience might be more familiarized with. You might know a company called Autodesk that started in the 80s and developed a product known as AutoCAD which basically allows architects to design and build complex mechanical structures much faster. Back then, AutoCAD was considered a super niche product with a target audience that basically consisted of architects. That's it, software for architects. Today, AutoCAD is still one of the main products of Autodesk, and the company is worth $50 billion. Then you have other cases like the semiconductor industry, also a very large market, roughly of $600 billion. And within that industry, a new industry emerged, known today as the electronic design automation industry, or EDA. And EDA, is led by a few companies such as Synopsys, which was founded a few decades ago. And Synopsys basically is helping electronic engineers to design integrated circuits. That's it, software for designing circuits. And again, today Synopsys, I think last time I checked, the company was also worth like $50 billion. So in a similar manner, and thanks to the development of what I was referring to, biotechnology 3.0, a new industry I see that is emerging and that will be led by a handful of companies. And some people have referred to this new industry as biological design automation industry. And these companies will be developing software for designing and building optimized biological products. Some people still, they argue that the market for biological design automation is small, but if you look today at the number of biologists in the US, it is as large, as the number of architects, and they use practically very few design tools. They've been doing everything for a long time using spreadsheets and then lab notebooks, and now they call them electronic lab notebooks, which is basically the digital version of the same thing. But things are starting to change, and it's not more about managing data, but understanding data and how to use that data to design new experiments and new products.
And I think it's been, it's amazing. It's starting to penetrate into biology, right? So many folks who became biologists, there's an old joke, they became biologists because they hated math. And I think pushing in some of that design software tools, those very mathematically savvy tools, that's been part of it. But it seems as though you're starting to get real penetration here as people see the simple utility of having an Autodesk-like design tool for biology. So can you tell us a little bit more about what Teslagen does, and what's the go-to-market, and how is that executed? What does it look like?
Sure. So, our mission is to help biologists and biotech companies to design, build, and optimize different biological products. And for that, we're building a very specialized software platform and AI algorithms that can help these biologists really speed up the design and build process, much faster, at least as compared to current alternatives. And I think the key difference here, maybe coming back to the analogy that I was making, is that when designing a mechanical or an electrical product, unlike designing a biological product, for a mechanical or electrical system, you have well-defined differential equations that you can solve and you can simulate the properties of your design. With biology, it's much harder. So our paradigm basically is to empower biologists to take a more iterative and high throughput and very data-driven approach, an approach where AI can also be used to train models and reduce the time it takes to find that optimal design or process. Today, the platform that we've built consists of 4 important modules that we call the design module, the build, the test, and the discover modules, in some way mimicking the so-called DVTL cycle, the design, build, test, learn cycle, where you are first designing the experiments, then defining and executing the experimental workflows, then gathering the data that characterizes your experiments, and then finally using AI to map the genotype to the phenotype of your experiments in order to guide the next round of experiments. And for us, what we've learned is that it's also important to communicate, very effectively with a series of, external tools and third-party services. So our platform communicates with lab automation equipment. It communicates programmatically with important companies that supply DNA, and importantly, it communicates with third-party databases so that you can inject also data from the outside, data that can be used to train AI models. And that's why we're like thinking that at least we call our platform an AI-powered operating system rather than a software platform, because interoperability is also key.
So can you expand on that's a very interesting point. Can you expand on that a little? So what are the aspects of an AI algorithm, or in general a discovery science workflow, that are effective for you, that are useful to you?
You mean I don't know if I kind of like maybe you can rephrase the question.
Sure. So in the properties of an AI algorithm that you apply, are you applying deep neural networks that are good at prediction but black box, or do you need insight as well? What kind of algorithms are useful in your
Yes. No, that I see. Yeah, that's a good question. So I mean, there are like a number of advancements in AI, specifically driven by the deep learning community recently. So like something that we've been looking into, and not only ourselves, but the community that is thinking how to apply AI to synthetic biology, are techniques coming from the natural language processing or NLP community. These are techniques that have been used by companies, for a long time. Companies like Google to power a number of their own tools. Now the instructions, that are written in DNA, to program life and to program a cell, of course, are not written in English. They are written in a different language that uses a different alphabet. Instead of using 26 letters of the English alphabet, we use 4 letters that encode different nucleotides. But similar techniques that have been used, very, successfully, in the deep learning community, that use recurrent neural networks, LSTMs, and so on, can now, for example, be applied to genomics to understand the language of life and to guide new experiments. So that's kind of like one example of things
that's very helpful. And now, could you make that concrete? What are some examples, some companies or applications where you see AI being applied? People you're working with, or fascinating examples from the community.
Yeah, sure. So I would say, at the high level, I would classify these companies or applications into maybe 2 large categories. Some of them are companies that are using AI to optimize the specific design of a byproduct. Usually optimizing a single stretch of DNA, encoding, for example, an antibody, or encoding an enzyme or a peptide. The second class or category of companies and applications are companies using AI and synthetic biology to optimize a whole microbial strain or cluster of genes, typically with the goal also of optimizing for the manufacturability of the product, which is also very critical for the industry. So, for example, not too long ago, we collaborated with the Berkeley lab and the Technical University of Denmark to optimize the production of tryptophan in yeast. So within one round of optimization, we were able to basically almost double the productivity of tryptophan in yeast. This was published by the Nature Publishing Group. And here, like what we did, to maybe go without going too much into the weeds, but we defined a design space composed of 5 key enzymes encoded by genes that are responsible for producing tryptophan. And what was under our control was the quantity, how much of these enzymes was expressed. And this already gave us, we were like playing with 6 different choices of promoter strengths, how much we produce of each one of these enzymes. This already gave us kind of like a design space of at least 10,000 possible designs. And then with machine learning, we started gathering some data. We could only do a few hundred experiments in each iteration. So we gathered some data, we trained a model, then we recommended the scientists to perform another 200, to build another 200 designs, and then demonstrated how very quickly we could optimize the productivity of this metabolite in high-value metabolite in yeast. There are other examples I was mentioning, Lansatec, that they are also optimizing the productivity of ethanol and other high-value molecules in some anaerobic microbes. But there's like, a number of examples. For example, coming back to deep learning, there are a number of generative deep learning models that can be used to do drug discovery. So we've in a project, we trained one of these deep learning models to discover novel antimicrobial peptides based on the known sequences of roughly a few thousand of these peptides, and then train the model to generate a new library of antimicrobial peptides that, with the goal of, in this case, optimizing the antimicrobial activity of the peptide. So there's a number of examples, but maybe those.
Well, that's a very beautiful example, because the drug there is a peptide, right? So you know how to manufacture the molecule from the genetic code. You know how to manufacture the molecule. What if I have one of these fancy new AI frameworks for drug discovery, and it's recommending completely novel molecules to me? How feasible is it in the near term that I could come to you with a molecule that's never been synthesized? Could you help me to design a platform for synthesizing a novel molecule? It's novel chemistry, or is that still science fiction?
It's not science fiction. Like, I think the capabilities of like these algorithms and the coupled with like all the molecular biology techniques that I was referring to, kind of like now allow us to start dreaming of exploring much larger design spaces, and these design spaces are going into directions that we didn't think before. So right now I was talking about these antimicrobial peptides, which are short stretches of amino acid residues, and there are 20 of these residues found in nature. But now pharmaceutical companies are exploring how to use synthetic residues, amino acids, artificial amino acids, to come up with peptides that are not even composed of natural residues, if that makes sense. And there's ways to encode the design of those molecules. This kind of crosses now the bridge from what is known as bioinformatics to chemoinformatics, but it's kind of like a close, cousin of, bioinformatics and computational biology. So I think eventually computational biology and computational chemistry will continue to kind of somehow merge, and both of them powered by AI.
So we're both based out of the Bay Area, so we can't talk about synthetic biology without talking about the CRISPR platform. Recent Nobel Prize to Jennifer Doudna. How has CRISPR and that new tool set, how has that impacted synthetic biology and your capacity to develop and execute experimental designs?
Yeah, that's a good question, and I'm maybe assuming that maybe the audience knows very little about CRISPR. Basically,
give us an explanation.
Yeah. So back then, when biotechnology emerged a few decades ago, it was key to something that the early biotech companies started using were, these molecular scissors called restriction enzymes, and these other enzymes called ligases to very efficiently cut and paste DNA. The thing is that these restriction enzymes that have been used by the industry for the past several decades, can only cut DNA at very specific locations within a DNA, stretch of DNA. And now came, CRISPR-Cas9, that basically, they discovered that you can program some of these molecular scissors to cut DNA specifically where you want, pretty much, where you want to cut, and very efficiently. So now people, that really was like a breakthrough. People started editing DNA through, CRISPR coupled with Cas9 and other enzymes in ways that were, not, imagined, for many decades. And they started extending these techniques to not only cut DNA, but also to program which specific, genes you want to transcribe. And there was a huge, huge revolution. This, as you mentioned, led to a Nobel Prize in CRISPR. And we've seen initially it was, adopted by the academic community. But more and more we see companies also they are starting to use it. There were a number of years where it was not clear who owned the IP rights of this new technology. There was kind of an IP fight, but now basically the things are settling, and there's a lot of, we see a number of partners and customers using CRISPR increasingly.
So I think it's fair to say that Teslagen was the first pure AI software company in synthetic biology, and still one of the only. It's a fascinating position. You're in an industry where almost everyone is very wet lab focused, and you're producing design software. Some of the components of which, especially the AI components, rely on access to big data. So how does your go-to-market strategy work around data access? Where does your data come from, and how do you build these incredible models that learn how to engineer biological systems?
Yeah, that's a good question. So first, we work very closely with our customers, and partners, and basically, given that we are developing the whole, platform for designing, building, and optimizing biological products, we are becoming their, if you will, also IT backbones. In order to bootstrap our partners, efforts, we've been using some techniques from the AI, community that people refer as transfer learning, where we basically pre-train some machine learning models using large, many times publicly available datasets to train what are known as encoders and decoders. And then we use our partners' proprietary smaller data sets to leverage on those pre-trained models. So that's number one. We also, given that our platform also lets our customers streamline the design of experiments and the execution of these experiments, then basically we also, if they have very little data, now we have the tools and the infrastructure to let them run experiments and gather data much more effectively. And then when they gather the data, that comes back to the platform, it's parsed and linked back right away to the original designs and the original experiments. So then you are pretty much ready to train a machine learning model, because you have the designs and the experiments, and then you have the outcome of all those experiments that are linked to the original designs. So you can then take advantage of all the infrastructure, for AI that we've been building. And really, our strategies, unlike other companies or, contract research organizations, we really want to empower our customers so that they can plan their own experiments. They can, we can empower their creativity and let them own also their, high value data sets. Ultimately they are the domain experts, and we're basically facilitating the platform, if you will.
So in the world of Ginkgo Bioworks and Amiris and these other large synbio design companies, what really makes Teslagen unique?
Yes, so like I would say, the first thing you were like talking about, Ginkgo and Zymergen, and there are other number of other CROs, contract research organizations, where they would try to do the experiments for you, for the company. We think that definitely we see that there are companies that outsource part of their efforts, but they still want to have control of a number of experiments that they want to design and build in-house. So one of the unique things is that we are basically empowering, as I was saying, these customers to design and run their own experiments. But in terms of like the technology itself, I mean, there are a number of tools out there. Some of them have been, developed by the academic community. There are some specific tools that, have been developed for very specific tasks, like tools for doing, what is known as codon optimization, or predicting ribosomal binding sites, or predicting metabolic fluxes, and so on. Our platform has been architectured to design and build bioproducts, either DNA molecules, protein molecules, microbial strains, at a much higher throughput than the conventional tools. When you design a molecule, as I was like explaining previously, you don't want to design one single molecule. You want to design a complex combinatorial library, maybe of 1,000 different molecules, and then you want to build, as many of them as possible, many times through automation. So what we're really focused on being very good running high throughput workflows, but also secondly, we've kind of developed a solution that allows companies and partners to design, build, and optimize your biological product from start to finish, and close, the so-called design, build, test, learn cycle, through the use of AI models, which is also something kind of quite unique. And in this process, we interface with other like niche tools and databases, but always kind of with the goal of like overseeing the whole flow of information, from design to build to test. If that makes sense.
It absolutely makes sense. So earlier today, in a session with Dan Jacobson on AI and computational biology, we talked about the notion of a self-driving lab, right? And that seems to me to be sort of the ultimate genesis of the design, build, test, learn cycle, that you have something that is very automated and pushes itself inexorably toward discovery. How close do you think we are to that, and how strong is the automation of experiments right now that might actually facilitate something like a self-driving lab for synthetic biology?
No, I think we're definitely getting there. I think that's kind of like the coming, in the next 5 to 10 years. There's a number of like lab automation equipment and platform, and that are letting you like automate significant fractions of the pipeline. Of course, like not everything is automated, and depending on the scale, like larger companies might have access to more automation equipment, and smaller academic labs or startups, for them is more challenging. But eventually, I think everything will be could be, or most of it could be automated, from, grabbing test tubes from a freezer to, putting it on plates and moving aliquots around and running different assays and gathering data. I think eventually everything will be automated, and also there's, like efforts to shrink, the size of the equipments and the amount of the volumes, of, liquids that you need, so that the automation process can be managed more effectively. So a number of like microfluidic platforms are emerging. So
That's extremely cool. So over the next, say, 12 months, what are the big milestones that Teslagen is playing for? What do you see as, what are the immediate big ticket action items?
For us, for the next 12 months, yeah, like, I mean, there are a number of them, we are going to keep integrating with the community. Recently, we announced indeed like a partnership with Biomate, this institution created by the Department of Defense. So that's kind of like an important kind of project for us for the next 12 months. There will be over 100 institutions in the US that are going to have access to Teslagen, and we're developing new standards so that we can empower the community to exchange data more effectively, particularly data coming from fermentation campaigns. Then there are, of course, like challenges that we're like dealing on the AI side of things. So there is as I was saying, one of the fundamentally, when you're like thinking about using AI for synthetic biology, the challenge is, dealing with a very high design space, design space with a very high dimensionality. And so we're going to keep exploring different like techniques to train different encoders so that we can deal with this, high dimensionality, fundamental kind of like challenge.
We should connect you to Pattern Computer while you're here, as far as fascinating companies that have come out of the Fire Conference. That's extremely cool. So can you, we're almost out of time. Just a few more minutes before we go to questions. Can you give us a sense of how large do you estimate the market to be where Teslagen is playing? I mean, what is the size of this market? Is Teslagen the next Microsoft? Give us a sense of the scope.
Yeah. It's first, like we estimate that, looking at like different numbers, we think that approximately up to $2 trillion, and this is, trillion with a T, not a B, $2 trillion are lost annually due to R&D inefficiencies. The question is, how can we, and this, this could be, reduced by, as I was saying, kind of speeding up the process of designing a new biological product. The question is how then, after reducing the costs and time, you can capture some of that value, in the process of doing so. Our, model so far has been pretty basic, has been, to simply license our technology to our partners. But given the growth that we see in the industry, the number of new biotech companies that are emerging at the global scale, the rate in which we see these biotech companies, adopting, synthetic biology workflows and adopting AI, and how the industry is moving, from using simple tools to more advanced solutions. Definitely, this is, this industry, what some people call biological design automation, will represent a multi-billion-dollar opportunity. As I was saying, the biotech industry is already large. As it happened with EDA, I think that maybe 2%, 3% of biotech will be captured by this industry. So it's already, like a large large multi-billion-dollar opportunity for us as Teslagen. For us, we're also exploring and actually closing a few deals where we're not only licensing the technology but also partnering with other key stakeholders in the ecosystem to get royalties based on the value that we add. This is kind of like another complementary strategy where we expect to have cash flows in the significant cash flows in the long term. But I think there's kind of in the last 3-5 years, there has been such a growth that finally there's an opportunity and a new industry that is emerging that is capturing the attention of the investment community, VCs, private equity investors, and so on. So I'm very excited about what's happening for the industry and also for Teslagen.
That is very exciting. I have seen such cool projects come out of SynBio. There's a little company in Emeryville, California, that has cloned spider silk genes into silkworms, and they make spider silk backpacks that are incredibly durable and stuff like this. It's been suggested in past years at FIRE that creating durable materials would be incredibly important for fighting climate change. And whether that's construction materials or spider silk backpacks, what do you see as the role of Teslagen in climate change, whether that's biofuels, durable materials, whatever it is? What do you see that as part of your mission?
No, definitely. Like as I was saying earlier at the beginning, I think a number of products that are derived from fossil fuels, as the primary source of the chemical pathway to synthesize those products, that is now being replaced by microbes that are used as small biofactories that instead of relying on these fossil fuels, these are microbes that eat sugar and produce a biofuel, or produce, as you were suggesting, like novel materials. And biologists, there's like so many cool things, products, materials that are already like produced by our by life, by spiders. So people are identifying the genes in spiders, for example, that are responsible for producing silk, and are copying and pasting those instructions into microbes so that you can produce them at a large scale, by growing yeast or growing E. coli
without having a swarm of spiders.
Right, without actually that companies, we were when we started Teslagen, we were being incubated in the same building where this company started. And I used to go down, I think they were on the 3rd floor, we were on the 4th floor, and they literally had spiders initially, like on the roof, this was the Genentech hall, that you see UCSF.
Well, with that horrifying image, Eduardo, we thank you so much. It has been such a delight to talk with you. And I will now turn it over to Evan. I see we've got a bunch of very interesting questions from the audience.
Yeah, I think, thank you, Ben. Thank you, Eduardo. That was really interesting. I think I've been to a few places that might count as a spider farm, but not in that way. So Greg Brando, I think you had the first question here. Are you with us?
I am. Hey, many years ago, Bill Joyce infamously said that, the world was going to be turned into gray goo by these things that we're unleashing. And I assume that there's smart people out there that are thinking about this. So why should we not be worried about building new things that escape and turn us into gray goo?
That's a good question. So there is, with any new technology, there are risks, advantages, risks, and definitely, risks have to be managed properly. There's like, different concerns, from the community, around, companies designing and building, bioweapons. It's not, I mean, there is, of course, there are like things that, technical barriers that, that even ourselves we've been implementing. So for example, we're interfacing with a few different services where, if a customer is, designing and synthesizing DNA. I mentioned that, our platform communicates with DNA synthesis vendors, other companies like Twist Biosciences, and the molecules that are being synthesized, we can screen them to kind of like see if there's like any, threat of, any bioweapon, that might want to be synthesized. And there are some protocols we would inform the FBI and so on. The other people are worried about bioethical issues. We can start like eventually, we can be messing with the human genome itself and doing experiments. And this has happened, in China a few years ago. So, there's as we develop these techniques, these technologies, they are very powerful, but also there are some risks, and there needs to be, also part of the community has to be thinking about these, ethical issues, which are extremely important.
Great, thanks.
Thanks.
And Mark, do you want to ask something?
Well, I will just mention that I think many of us are friends of Bill or were, and he was going through a very tough time in his life when he wrote about the end of the world and the things that would cause the end of the world. I think that he's not so dark now.
Well, that's nice to ponder, actually. Yeah.
I think again, there's like a number of like risks that need to be managed properly, but eventually, like the benefits of like moving to a bio-based economy are so, large. They, outperform in my opinion, so much eventually the risks that definitely it's, something that we have to keep working on. And on the like bioethical, side of things, I do think that there are a number of diseases that can be eradicated with the use of synthetic biology, particularly, diseases that are known as Mendelian diseases that are caused by a single mutation in your genome. So there's a number of companies, biotech companies and pharma companies that are developing different gene therapies and cell therapies that are, really exciting to see, and eventually, maybe in the next 10 years, 20 years, like even, things like cancer might be tackled with biotechnology. As we understand, there's a strong amount of evidence that, there are genetic causes, driving the, that generate cause, a number of different cancers. So as we understand more, and we're going to be curing a number of diseases and moving into a bio-based economy, but we have to be, we have to do it very carefully, and manage risk very carefully.
Evan and Eduardo, and I would put this a different way, that the stuff that Michael Crichton is afraid of, is usually the wrong stuff. But great author, good movies. But if you want to worry about something, don't worry about advanced general intelligence. Worry about something much more close to home. FAA planes crashing into each other, kind of a thing. So I think that the question of, which I personally believe is a legitimate question, was COVID man-made or some version of COVID? We know that we Americans made a kind of a version of that in North Carolina. We know that we shipped it to China. We know this. We paid for it. We don't know if wasn't exactly the bug that got out. I think we're pretty sure now that it was a lab release, despite the recent propaganda from China again. So okay, but it could have been accidental. But I think what you're addressing, Eduardo, is it's not that hard for a rogue group of smart people to make a bioweapon, and that's dangerous. That's really dangerous. So they wouldn't have to use your tools, but they could do that kind of thing. So that's a threat to the world. And if we had 10 COVIDs right now instead of one, could we get through that?
We have 4 minutes.
All right. Okay.
So I'd love to get some more questions. Ricky had 2 questions, one of which might be eminently relevant. So Ricky, do you want to kind of just ask those both at the same time, and then maybe you and Eduardo can connect if.
Yeah, the first one was already answered, which was the concern about Wuhan. But the second one, I was wondering, Eduardo, are you open for investment? I'm really fascinated by what you're doing.
Well, as an entrepreneur and currently CEO of the company, we're always, interested in talking to investors. So yeah, if you want to connect, happy to connect. I think my email is in my profile page on the website. But we can talk more. Yeah.
Fantastic. Bobbin Teagarden had a question about CRISPR. Bobbin, are you with us?
Yeah, I was going to ask more technical questions, like how are you doing this? And one was CRISPR. Is CRISPR like a CAD/CAM or BioCAD system that you use up front to do what you're doing? And then what AI are you using behind it? And are there one or more ontologies with the knowledge structure of what you're doing?
So if I understood the question, what is it specifically that we're doing around designs and experimental workflows that involve CRISPR? And there are a few different things. As I would say, we have different modules that cover the whole design, build, test, and kind of learn cycle. There's a number of companies on the design side of things, and build side of things before running the experiments and capturing data coming from these CRISPR experiments. We've seen that, and particularly given our strengths and where we see our kind of what our paradigm is, we see companies that are using our platform to do some knockdown or knockout experiments using CRISPR, but they are not targeting one gene at a time. They are, creating large combinatorial libraries of, say, CRISPR knockout experiments. And so using Teslagen, you can really design these experiments very effectively, very efficiently. There's still, when you're people, maybe I'm getting more technical, but typically when you're designing a CRISPR experiment, you have to design some what are known as some guide RNAs or some homology DNA sequences, homology arms, and all that can be automated and can save you a lot of time, particularly when you're thinking about doing 1,000 CRISPR experiments in parallel. And then, the nice things of, our platform is that after designing, the design instructions of your experiments kind of go to the next module seamlessly. So then you want to run the experiment, you basically import the design instructions from the design module and execute your experimental workflow very kind of in a very streamlined manner. And then depending on of course, the nature of the experiments, companies want to characterize the effect of these, let's say, CRISPR knockouts on the, there's always an end result. Maybe they might be trying to optimize the productivity of a metabolite or a molecule. That's when, after gathering the data, they feed it back to the platform and train some machine learning algorithms to kind of guide the next round of experiments. There are other opportunities to using AI, specifically in CRISPR, that we've seen that we've been exploring that are more specific to CRISPR, which is many times when you are designing a guide RNA, there's also some collateral effects of your experiment. You're intending for this Cas9 enzyme to knock out the gene in a specific region in your genome, but maybe your guide RNA might be also having an off-target effect. And so there has been some needs by the community to learn how to minimize those off-target effects, and kind of like understanding how different Cas molecules work and what are their target effects. This is something that you can train with some AI models. There's a lot of work done in like model species like E. coli, but there are a number of companies that don't use the model species, that use some other more non-model species where there's less data and where they need to maybe use AI to characterize the off-target effects of Cas enzymes, if that makes sense.
Awesome. Thank you, Eduardo. And with that, we are out of time. Thank you for a fantastic panel. We appreciate the time you spent with us today. I know you have probably a lot on your plate, so it really means a lot to us when people come and